What's up, W Gang? Today we're talking about ATP synthase and how it works in the life around us and especially in humans.
Matt, first question, what is the ATP synthase gene's job in humans?
So a gene specifically creates ATP synthase, which is made out of multiple proteins, and that gene produces and transcripts those proteins into the ribosome. So once it's done, the ATP synthase comes out and is at the cell membrane, mitochondria, and chloroplast, so that it can move the hydrogen gradient, and while that's happening, it's creating ATP. So, I also have a question for you. If do all organisms with this gene make the same protein?
No, Matt, not all organisms with the ATP synthase gene make the exact same protein. The shape and structure and function of the protein depends on the type of cell it is on and the environment within the cell, outside the cell. And obviously, within different organisms, different species, the environment outside the cell is going to be different. My question for you is if this gene appears across many species, which we know it does, what does that say about evolution, Matt?
It just says that the gene is the gene and ATP synthase itself is very ancient, and it's a fundamental enzyme that is needed in cells and other prokaryotes. It likely originated early in the evolution of life and is essential for energy production, which means without this, many species today would not be alive.
I agree, Matt. To add on to that, ATP synthase is the primary mode that species and organisms and cells get its energy, which means that must mean that ATP synthase evolved very early on in evolution, because without it, energy would be a lot more difficult to come by. Also, in addition, the endosymbiont theory says that at one point, multiple prokaryotes that were mitochondria and chloroplasts merged into one cell. Before that, energy was mainly consumed and produced just by consuming straight glucose molecules from the environment, until ATP synthase evolved, which helped use glucose to make even more energy.
So I also have another question. How do AI tools complement the traditional genetic analysis?
So AI shortens the time and obviously shortens the expense that is associated with genome sequencing. Tools like BLAST you can insert a nucleotide sequence from one gene, and then it will automatically and very quickly scan through its entire library of all gene sequences, all genomes, and try to find the matching sequences to that sequence you put in. That's just one example of an AI tool, BLAST. There's also other tools, obviously ChatGPT, another one for genes, AlphaFold. Those are very useful with finding very quick and accurate results for any question you ask, any question you have.
Yep, and
my question is, should we rely only on AI for these evolutionary studies, or do physical traits like morphological traits still matter?
So I would say we should not only rely on AI for evolutionary studies, because it isn't fully developed, and morphological data is also very important because you can tell the difference between a reptile or a mammal. And if there was an AI that was developed, certainly like in a specific area in the gene, like if an AI was developed just to look at genomes and genetic sequences and combine them, then maybe we could rely on AI more. But right now, AIs that are created are not specifically made for that. They're made more for questions and answers, not specific areas like engineering or genetic sequence.
And as with all areas of AI at the moment, there's way too much risk for incorrect results at this point in time. In this point of AI development, you could see that many studies have already been made on AI, on ChatGPT, on Gemini, on other AIs, on Grok, that just show that they've been known to give you completely incorrect information, which you do not want, especially if you're using it in a scientific study.
Yep. All right. See you guys. And
all right, Matt. We used a few AI tools in this project here, so I'm gonna give you a few of the names, and you're gonna tell me how many booms they get. So number one, we got AlphaFold, which was the one we used for like 3D structure, getting a picture of the protein.
I think AlphaFold is very developed in just a certain area, but it isn't fully, you can't trust it fully. So I'll give it 4 big booms because it's not the best.
That's fair.
So boom, 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 boom.
All right, that's fair. Next one, we got ABI, AI, and DeepMind. We didn't use this one too much, but it's the purpose of it is to just explore how the gene interacts in biological pathways.
So this gene is specifically developed in there. So I'll also give it 4 big booms because it's made for that purpose. Boom, 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 boom.
All right, next one we got Twitter's AI, Grok. Mr. W Batman loves to use Grok for some reason. I think he just likes
Twitter.
Spends so much time on X. But we'll see Grok. I know what I'd give it, but I'm curious what you would give it, Matt.
I'll give it. It's an alright AI. I'll give it 3 AIs. 3 big booms. Boom, boom, boom.
Just for the record, I'd give it 1 or 2, to be honest. Last time we got ChatGPT. I wonder what this one's gonna get.
So ChatGPT probably superior AI, research AI. You know, it's just very useful, and I'll give it 5 big booms. Boom, 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 boom. All right.
Thank you guys.
That's a wrap for the video. Like and subscribe. Comment what you learned, and see you in the next video. Goodbye.
Goodbye.